For those that weren't in the last session, I'm Matt Lara. I am the Exploration Project Scientist for Biological and Physical Sciences. It's a division within the Science Mission Directorate. So today I'm talking about microbiology or microbiome of the built environment, or MOBI, on Artemis 3. Next chart, please. So just to settle on some nomenclature here. Microbiology, it's the study of microbes. That, we all took that in college. Looking at organisms such as bacteria, viruses, fungi, etc. Microbiome is that the community of microorganisms that live in a specific environment. So this is really what we're focused on. That environment could be the human body, it could be an ecosystem, it could be a closed environment vehicle. So that's the built environment, which we're talking about today. And then microbial ecology is environment. It's also known as environmental microbiology, that studies the interactions between these microorganisms and their environment. So their environment, each other, other organisms. So we're looking at the microbiome, the built environment. So in a closed system, every time you walk into a room, you're impacting the microbes that are present in that room. So, and that room is impacting the microbes that are present on you. So it's important to track that and look at how those things evolve over time. And in Artemis missions, it's a semi-closed case where it's a semi-closed system where the microbes that are going to be present are brought with them by the crew, and every time there's a some kind of change out, that might change. We want to see how in a closed system or a semi close to closed system, how that microbiome tracks over time, changes, and that impacts things like biofouling of systems and things like that. The next chart, please. So the overarching scientific goal, which I kind of touched on the last slide, is to model a microbial ecosystem in a closed environment. So that's relevant to deep space, lunar planetary surface missions, as these impact the crew health, it benefits the crew health, and also the exploration systems maintenance. So there are stories of previous stations like Mir that dealt with some pretty significant biofouling. So we want to be able to predict that and mitigate for it. This is an area of research that's been pretty extensive on ISS. It's a definitely a unique case that there's vehicles that are going in and out of ISS pretty constantly. So that microbiome changes over time, and we want to know what that's going to look like and how to model for it, how to predict it, how to mitigate it for deep space missions. So the science of MOBI is essential to obtaining data for building a comprehensive understanding of that ecosystem. So again, how it's interacting, how it's changing over time, and how it interacts with the crew themselves. We want to be able to understand, characterize, and catalog the flora of a vehicle. How stable is it? Does it change over time? How does it change? What's present? How things evolve over time. And then understand and model that ecosystem and individual microbe growth. So, and how these things are changing in response to the spaceflight stressors that the microbes are experiencing. And then understand and model the microbial transfer between different surfaces. So as the crew's moving around, as vehicles are docking with each other, how things are interacting with each other, and how the microbiome changes over time again. And then again, that the engineering, how it impacts the engineering subsystems. So water storage, filters, the different subsystems can deal with biofouling. So we want to make sure that we're tracking what microbes are present, and do that consistently throughout the life of a mission. Next chart, please. So specifically for Artemis 3, what does this look like? So the overarching goal of MOBI on Artemis 3 is to obtain the seminal data of the microbiome of the spacecraft. So we want to look at it and do all the analysis in situ for this mission. So we're not reliant on sample return. We want to be able to do this so that in future missions they can do this on demand and constantly be tracking this. So the data obtained will inform and guide future more comprehensive studies, on future vehicles, future missions. So the specific goals are to survey and characterize the environmental microbiome of Artemis 3 in the Orion capsule itself. So the second specific goal there is to evaluate the operations of this MinION based system. So this is a swab to sequencer system that has been in development for ISS, and we want to make sure that it works and we can do the in situ analysis on a flight without having to rely on sample return. So there's there's multiple components that go into this. There's the swabs that you see there in this picture. So the crew will swab specific locations within Orion, and then put that into the next box there that's behind those swabs with the little knob on top next to the left of the iPad. That is a mini PCR machine, so that'll actually extract the DNA from the samples and get it into such a situation where you can actually process that sample in the MinION. So the MinION is a sequencer that is just looks like a little USB drive that's plugged into the iPad to the right. And so the crew will, once they have the DNA extract, they'll use the pipetter to pipette that into that MinION and hit go on the iPad, and that'll give you your. Looks like we have an open mic maybe. That then we'll get the sequencing data there. So that's the only thing that comes back is the sequencing data itself, and that's eliminating the need for sample return and any logistics associated with sample return. So it's a pretty complete throughput process that there's a lot of details that need to be worked out with. The chemistry that goes into the mini PCR machine needs to be optimized and done with safe chemicals that are all low hazard levels, and so that's all in development now. But it's going to be ready for Artemis 3, and we hope to use it on future missions as well. So next chart, please. So yeah, just a couple of acknowledgments here. This is really Sarah Castro-Wallace's baby. She's at JSC. She's the expert on MOBI, and this is her hardware and chemistry that she's been developing at JSC for different missions within HRP as well as BPS. So there's a couple other experts there that have gone into developing the concept for this, as Margot and Venkat. So I think that's my last chart. Yep, that's my last chart. So again, yeah, it's I was looking at the schedule for this and realizing that we have two competing names for this thing. We have microbiology of the built environment and also microbiome of the built environment. So I'm going to take the action to go back to Sarah and see which one we're settling on, so that we can get our titles straight going forward, get our opnom all worked out. But as regardless of the name of it, it's an important experiment, and it's an important field that we'll be doing from future missions, not just Artemis 3. Thanks.
All right.